Hello students, this is Faizan Mirza. We are discussing the topic of selection and evolution. Right now we are discussing the subtopic of evolution, that how natural selection leads to evolution. There are various selection pressures exerting at a naturally occurring population. There are four graphs you can see here. The first graph actually shows you number of population, which means the frequency in the population, and the x-axis shows you the body mass. This body mass is the mean body mass. You can see the mean body mass is most frequent in a population. Selection is occurring against very small animals and very large animals, the two extremes. This is continuous variation. You can see the largest ones, the heaviest ones, and the lightest ones. So the selection pressure occur, or you can say it works against these animals which are very large or which are very small, and over the time stabilizing occurs. And because of this stabilizing, animals with a very low body mass and a very high body mass are subtracted or omitted from the from that environment. And the you can see the graph shrinks at its edges. It is defined with new extremes here, and you can see that the mean stays the same. This entire process is called as a stabilizing selection. So this is stabilizing selection here, which means it keeps the things the way they are. This is how evolution occurs, or you can say natural selection occurs against animals which are more suited to a certain environment, less suited to a certain environmental condition. Now let's suppose, for example, if selection starts to occur against very small animals as well. So initially these were the ones who got subtracted from the population. Now let's suppose if the selection pressure is exerting against this population, this type of small organisms, not the smallest but very small, the larger than the smallest ones. So if selection again occur against them, so what will happen here? They will be omitted from the environment as well. So over the years, it will be seen that the graph will shift ahead, and this kind of selection is called as a directional selection. Directional selection actually shows to you that now the mean will be changed. The mean body mass, which was most frequent initially in a population, will be shifted ahead. It will be having a new mean, and that new mean will be the most common occurrence in the population as far as the body mass is concerned. So this is called as directional selection. Then another kind of selection can occur, which is called as a stabilizing selection. The disruptive selection. What is disruptive selection? In disruptive selection, the selection occur against the animals of middle size, these ones which are neither very large nor very small. So in this case, number one, you can see the extremes are very well there. You can see that the frequency in population of this mean body mass, as compared to this, it has decreased. So the organisms are less in number with this particular body mass, and the ones which are there are having more frequent body mass of these two sizes, this one and this one. And you can see that the peaks are actually lower than the ones that we have discussed initially. So this actually tells you that there will be two, there will be populations having two kinds of peaks here. This value will be more frequent, this value will be more frequent as well, but the one which was the mean, which was the central value of this particular variable that you took on the x-axis, will be less frequent in a population. This kind of example can be seen if you exemplify this for your understanding. So this can be seen for fishing practice. So when fishing is being carried out, normally fishes with a middle body mass are the ones which are fished out by the fishermen, and these are the ones with the highest market value. So fishes which are very large, fishes which are very small, they are not taken because they don't give good worth to the fishermen. So, and as well as the demand. So this is why the selection occurs against these ones. So although these were supposed to be the highest in the population according to the stabilizing selection, but because of the selection pressure occurring against these middle-sized animals here, which in this case is overfishing of this kind of animal, so this got decreased in this population. This is called as disruptive selection. This example here, you can see percentage of population and the birth weight in kgs is also shown to you. You can see that the mean birth rate, the most common birth weight in which the percent of population falls, is the one which is in the central value. You can see it's also a kind of a bell-shaped curve, and bell-shaped curve with the extremes, and the extremes are very low in population. The central value is the highest one in the population, and if you just convert it into mortality, so this line actually shows you your mortality rate. You see that that babies with a very low body mass are likely to die. Babies with a very high body mass are likely to die as well. But babies with a very medium body mass, a mean body mass, they do survive. So this is also a kind of a stabilizing selection occurring here. Another thing that we are discussing here is natural selection. Some examples here. So if we take an example of Bitsin Betullaria, this is the peppered moth. When we write the names, we use the binomial nomenclature. This first word here, Bitsin, it represents the genus, and Betullaria represents the species. This is how, for example, we are Homo sapiens. We write Homo here, we write sapiens here. Human beings are Homo sapiens. So genus of this peppered moth is Bitsin Betullaria. The first name is written with a capital letter. The second name is written with a small letter with a gap in between, and you underline both independently. This is how we write the names in binomial nomenclature. So for example, if we take an example of peppered moth exists in two forms, two phenotypes: white peppered moth as well as black peppered moth as well. White was more common before Industrial Revolution, and the black one, it became more common after Industrial Revolution in various parts of the world. Why was that so? So that actually was because of the waste gases that killed the lichens which were actually growing on the tree barks. What was lichens? Lichens or lichens, either way you can pronounce it. This is a kind of white fungus which grow on trees. So this peppered moth, it rests on the tree during the daytime, and when it's resting, it's likely to be caught by some kind of predator. So you can just see here, this is the black peppered moth sitting against the lichened trees. These are the lichens grown on the tree, and this is the bark of the tree. Because of this lichen, you can see this is the white background. The black one stands out very well, but if the tree trunk is bare, it's not having any lichen, so black one gets camouflage well. So if this was the case before Industrial Revolution, the barks were covered with this white fungi, which is the lichens there. So when during the daytime, the white phenotype, this peppered moth would rest, and the black would rest as well during daytime. Black one was likely to be caught by the predatory birds, because the predatory birds would use their eyesight for seeking their prey, and they would spot this black very well against the white background. But after Industrial Revolution, the lichen, this white fungus, got killed because of the waste gases, so the background turned black, and now the black one got it got advantage here, a selective advantage here, and the white one would when the white phenotypic variant of this peppered moth would sit on this bare trunk during the daytime. The predatory birds are likely to catch that white one on this trunk, because it would not get a very good camouflage. So as a result, the black one after Industrial Revolution was likely to grow adult and survive and pass on its genes to its offsprings and reproduce. So a large population became black. And before Industrial Revolution, a large population of the peppered moth was white and less was black. This was because initially the environment was favoring the white one, and now the environment is favoring the black one. So the allele of black and white always existed, but it is the selection pressure that initially the selection pressure was against the black and it was selecting white, and now it is against the white, it is selecting the black. Various examples exist for bacteria as well. If you just can recall E. coli and Plasmodium, the malarial causing bacteria and the Plasmodiums. So you can see that here we are also using the same kind of binomial nomenclature. For example, Vibrio cholera, we write V with the capital V, Vibrio, and cholera with a small c, and we leave a gap here and we underline them separately. And Plasmodium ovale, Plasmodium vivax, Plasmodium malariae, Plasmodium falciparum, we always use the binomial nomenclature. And for example, if we are using a symbol here, or you can say not the complete word but a letter, so we write E. coli, rather than Escherichia coli, we write E. coli. So E must be capital with a dot, and we underline it, and coli is underlined separately. And keep in mind that the first word, or the first word in this binomial nomenclature, it represents the genus, and the second one represents the species. So these are the malarial causing agents. They all belong to the genus of Plasmodium, whereas the species is ovale, vivax, malaria, falciparum, and similarly Escherichia coli, E is the genus and coli is the species. If we discuss antibiotic resistance, we know bacteria have a single allele only. They do not have homologous chromosomes. They always have a single allele for a gene. So a qualitative difference is there because of the presence or the absence of that single gene. And that can give a huge selective advantage to that particular bacteria who is having a gene or who's not having the gene would be at certain disadvantage. For example, the bacteria with resistant gene, resistance against any antibiotic will always be minor in population. It would survive the antibiotic. It is not killed by an antibiotic. So if for example, if you are using antibiotic, what the antibiotic would do, the antibiotic would kill all the bacteria who lack that resistance. So the bacteria that lack the resistance, which form the majority of the population, they get killed, whereas the minor population survives, which had the resistance gene with them, so they survive. And when they survive by the use of or overuse of antibiotics, what do? We kill the major population which lack resistance, but the minor population which had the resistance against that antibiotic, it survived. Why they had the resistance? Because they had the gene, the particular allele that could give them selective advantage, and they could survive that kind of antibiotic being used. So this minor population, once this major population have been wiped out, this minor population gets access to 100% nutrients there, and using the 100% nutrients, after some time it will form the 100% population. So initially, although the nature selected them in such a way that a minor population was having antibiotic resistance, the major population was not having antibiotic resistance, but the overuse of antibiotics kill the major proportion of the population, and then the minor population which initially is having the resistance gene, so although they were a minor population, now they have access to all the nutrients, all the conditions in the favorable condition in the environment. They are not facing any competition with the major population anymore. So after some time they will form the 100% population. So this is how antibiotic resistance gets promoted because of overuse of antibiotics. These agar plates, this is agar grown here, and this agar is actually having bacteria inoculated on them. You can see the bacterial growth. This orange stuff is the bacterial growth. These white ones are the circles of disc which are impregnated with a certain antibiotic, and they are kept in the lab overnight or for some hours in the incubator, and after that you just go and you just see a clear zone around certain of these impregnated discs, and not all. So what do these cleared areas indicate? This indicate that the bacteria was unable to grow in this region because this antibiotic is effective in killing this bacteria, whereas this antibiotic, which is a different antibiotic, another different antibiotic, so they were not able to kill this particular bacteria. Similarly, this is some other bacteria and some more. For example, you can see bacteria which are non-resistant, they will be easily wiped out, and bacteria which are resistant, they will not be wiped out. This is where you can see that this is the diffusion of antibiotic from that impregnated disc into the agar. That's why you get the circular area here, which is the clear zone. Now we discuss another topic, or you can say another part, which is the sickle cell anemia. An example of sickle cell anemia, there can be three different, there are two alleles for the hemoglobin beta chain. It could be either HbA or HbS, and a person could be either HbA, 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 HbS, or HbS, HbA. These are the three types of people, depending on the genotype that they can be having. In Africa, two selection pressures are actually working simultaneously on the same population. One selection pressure is malaria, the other selection pressure is sickle cell anemia. So people who are HbA homozygous dominant for this allele of normal hemoglobin, they die from malarial endemic. What is endemic? Endemic is the disease which is always present in the population. So they die from malaria. HbS, they die from sickle cell anemia because their red blood cells cannot, number one, give appropriate oxygen. And then these sickle cells, they tend to get stuck, or they tend to get stopped in the blood circulation somewhere, and they impede the blood flow, and then clotting can occur within that region, or internal bleedings can occur, and they die because of sickle cell anemia. Whereas people who are heterozygous for this condition, they do not die from sickle cell anemia, and plus they are resistant to malaria. So this is a survival advantage here. You can say it can survive both selection pressures. One selection pressure is malaria exerting on this particular individual. The other selection pressure, which is sickle cell anemia, is working against this individual. But the person who is heterozygous dominant will survive malaria as well as the sickle cell anemia. So over the you can say over the years, African population have evolved, and it is seen that now the majority of population in Africa are HbA, which means they are the heterozygotes of this particular genetic combination. And because they are the heterozygotes for this HbA condition, whenever they reproduce, this is the kind of Punnett square that we can expect: HbA, and HbS. So this is the outcome. What does this tell you? This tells you that whenever these two individuals, which are the majority ones in the population in Africa, whenever they reproduce, they will be producing any one of these offspring. We can just recall that since the selection pressure of the sickle cell anemia is working against HbS, so this is likely to get killed, whereas HbA is likely to get killed from malarial endemic, where so HbA and HbS should survive. And if you just go and recall that the bell-shaped curve that we discussed, so the stabilizing selection should omit out these individuals and these individuals from the population altogether. So HbS should cease to survive, and HbA should be omitted from the environment as well. The environment should only be having in Africa the heterozygotes, but that's not happening. Only the majority of population is HbA, whereas HbS, and HbA are still being produced. Why is that so? Because individuals who are there in Africa, most of them are actually HbA. So whenever they reproduce, this is the kind of Punnett square which I was talking about. So there will be 1 in 4 chances that this individual will be produced, which can die from malaria endemic. There will be 1 in 4 chances that an individual offspring can be produced, who is susceptible to dying from sickle cell anemia, and there will be 2 in 4 chances that the same kind of offspring will be produced similar to the parents. This is why malaria and sickle cell anemia, they have not been able to wipe out the HbA combination or HbS combination from the African population. And we discussed this initially, that the mutations are mostly recessive and they confer disadvantage. Here you can see it's conferring disadvantage as well. But this is an advantage. This is a huge selective advantage, that you can see that the presence of this recessive allele together with the dominant allele is given resistant against malaria, which is an infectious disease, and in areas where this is an endemic, this is a huge selective advantage, and people who are heterozygous for this condition, they will survive both selection pressures. This actually gives us a clue that this is how evolution occurs. This is how mutations, they accumulate in population over the years, over decades, over centuries, and these are the mutations that eventually confer advantage. You can see this kind of mutation is conferring advantage, but only in the heterozygous condition, not in the homozygous condition. And these people have selective advantage, and this is how this can give us a clue how evolution actually occurs over time. That's it for now. Thank you so much.